Okay, so then let's get started. So welcome to Environmental Research webinar. So I am Sungkyun Park, Associate Professor of Epidemiology and Environmental Health Sciences. And this webinar is sponsored by the Michigan Center on Life Stage Environmental Exposures and Disease, in short, M-LEEaD. And I am very pleased to introduce today's speaker, Dr. Douglas Walker. Dr. Walker is Assistant Professor in the Department of Environmental Medicine and Public Health at Icahn School of Medicine at Mount Sinai. He received his BS from the University of Massachusetts, Dartmouth, and completed his PhD from Tufts University in Environmental and Water Resources Engineering. During his postdoctoral fellowship at Emory University, he acted as director of Exposome research for the Clinical Biomarkers Laboratory and was a member of the Hercules Exposome Research Center. Dr. Walker is currently head of the high-resolution Exposomics lab in the Institute for Exposomics Research at Icahn School of Medicine at Mount Sinai. So please welcome on joining. So Dr. Douglas Walker.
Great, thank you. And thank you again for the opportunity to present today and share some of the research that we're doing, attempting to improve our ability to characterize the exposome and how environment contributes to disease outcomes.
So you need to switch the, so now we are seeing the, yeah.
Okay. I thought we fixed it last time, but how's that? What do you see?
Yeah, now it's good.
Okay, perfect. Thanks for that. So just as an overview, our research is focused on developing methods that allow us to apply omic scale exposure assessment and epidemiology of human disease. We work in a number of different populations and study systems, but our ultimate goal is to develop analytical methods that allow us to better characterize biological samples for the presence of environmental exposure biomarkers and biomarkers of effect associated with those exposures. We primarily focus on cancer and metabolic disease outcomes, but we've worked in a number of different disease systems and disease outcomes, and have really found that these methods allow us to improve our ability to characterize exposures in human populations. So within our laboratory, we use untargeted high-resolution mass spectrometry within an exposome framework to link three separate pillars or focus of our research program. The first is using these untargeted methods to better understand environmental risk factors of cancer, specifically blood and lung cancers. We also apply these methods for bioeffect prioritization. So we use different bioactivity and genotoxicity assays to try and identify which exposures could be driving cancer risk. And finally, because these analytical methods are very powerful and allow us to characterize samples to depths that weren't possible previously, we also leverage these methods for understanding emerging exposures of concern. And primarily our work right now is focused on establishing biomonitoring methods for microplastics in human populations. So during my presentation today, I'm going to really just touch upon three different areas. The first will be to really provide a practical definition of the exposome and how we're incorporating that into our research. Next, I'll focus on how we're using high-resolution mass spectrometry methods to better measure the human exposome. And finally, I'll give a very brief overview of how we're applying exposomic strategies to measure microplastic exposures in human populations. So following completion of the human genome, it was anticipated that our ability to understand and predict disease risk would greatly improve. We've seen tremendous advances in genetic technologies and genetic testing over the past few years or past 20 years. However, what we found from a lot of our research is that genetics alone cannot tell us all that we need to know to predict disease risk. And this is evident when we consider population attributable fractions for many common chronic diseases. This chart is from a paper that Steve Rappaport published about 5 years ago now that used different studies of monozygotic twins to try and calculate the population attributable fraction when you account for gene or gene by environment risk factors. And this population attributable fraction can be considered the proportion reduction in a disease if you account for these genetic risk factors. And what he found in his paper that for most of these, when you consider these genetic risk factors, you're only reducing the disease by about 20 to 30%. The remaining prevalence of the disease or the remaining variants of the disease risk factors cannot be ascribed to genetics or their interaction with environment. So what really drives our research is trying to understand that other component and measure that other source of disease risk. So our current estimates suggest that genetics or genetic predisposition for disease only accounts for about 5 to 15% of disease risk. The other 85 to 95% is derived from environment, lifestyle factors, and its interaction with the genome. So if we consider heritability alone, we're really taking an unbalanced view of human health and disease. So this need to better understand environment and its role in human health outcomes led Christopher Wilde to define this concept of the exposome in 2005. Since then, it's undergone a number of redefinition and evolutions to what we consider the current concept or current framework for the exposome, which is the cumulative effect of environmental exposures and the corresponding biological response across the lifespan. This definition is really key to the practicality of the exposome because it's not aiming to systematically measure every possible exposure. Rather, we're interested in the cumulative effects and how those lead to biological changes throughout the lifespan. And so as a whole, when we consider the exposome, really we consider any non-genetic disease risk factor as contributing to the human exposome. This can include things like physical chemical exposures, psychosocial stressors, and other factors that all contribute to our health and well-being. However, my laboratory and our research is really focused on establishing methods that allow us to better understand chemical exposures from a variety of different sources. While those other contributions to the exposome, particularly the psychosocial stressors, are extremely important and often interact with these chemical exposures, our methods are much better poised to measure the chemical contributions to the exposome. And so that's what I'll really focus on for the remainder of the presentation. So our challenge is really in developing approaches that allow us to characterize the exposome to the extent of its importance. So there's been tremendous advances in our ability to characterize the biological phenotype, the capabilities of whole genome sequencing and genotyping and gene expression analysis have really just become incredibly comprehensive and low cost over the years. However, our ability to characterize environment is really limited relative to its importance. And even some of our best targeted analytical strategies only allow us to characterize anywhere from 10 to 100 exposures at a time. So what we focus on is really trying to develop analytical frameworks that allow us to perform universal screening of the human exposome. So next, I'll go into more details about how we're actually doing that in our studies. So we've approached measurement of the human exposome through methods that allow us to characterize exposure biomarkers in biological samples, as well as biological response. And we do that through application of untargeted metabolomic methods that allow us to characterize a very large number of small molecules in the human body. There's unique advantages of considering metabolites in exposome research because it provides us two of the key measures that we need for exposome research. We can look for biomarkers and metabolites that arise from exogenous sources. These can include things like exposure to commercial products, environmental chemicals, and their metabolites. These also include things like drug and drug metabolites, as well as food constituents. Within the same assays, we also have the ability to look at endogenous metabolites. And this allows us to evaluate bioeffect and how we see changes in endogenous pathways related to exposures and disease outcomes. However, our current estimates suggest that in order to thoroughly account for a representative exposome and metabolome within a human biological sample, we have to have the ability to characterize tens of thousands, if not hundreds of thousands of compounds. To do this, it's really not possible to consider application of traditional methods where we first identify the analytes that we want to measure, develop the methods to measure those, and then measure them in our samples. Rather, we have to use approaches that allow us to prioritize which exposures we should be characterizing and let the data really drive which exposures we should be studying. So in order to really embrace and understand the complexity of the human exposome, it's critical to adopt analytical strategies and study designs that allow us to incorporate untargeted measures of exposure. When we're performing untargeted analysis, we are not selecting our analytes a priori, but rather we are developing methods that allow us to detect as many chemical species in a biological sample as possible. With these methods, we then identify the populations that we want to characterize and try to detect as many compounds as we can in samples from that population. Then we use biostatistics and bioinformatics to identify which signals and which chemicals we should focus on in terms of our health outcomes or whatever our study outcomes are. And an example of the advantages of applying these untargeted methods was really highlighted by our application of untargeted exposomic platforms to study occupational exposure to trichloroethylene. So trichloroethylene is a degreasing solvent that was very commonly used in manufacturing processes back in the 70s through the 80s. It's a very widespread environmental contaminant, and it's been known to be linked to different cancer and immunological outcomes. So in this study, we were able to collaborate with a team of researchers who was doing extensive monitoring of workers who were using trichloroethylene in a manufacturing process at a factory in China to better characterize, and our goal was really to understand early biological response to this exposure to potentially understand how it could be related to the cancer outcomes that we were studying. So in this population or in this study, our collaborators were able to identify 95 unexposed workers, and these were workers from factories who did not use any volatile organic compounds in the manufacturing process, as well as 80 workers who were using trichloroethylene, mostly for degreasing purposes or cleaning manufactured parts. Once these workers were identified, they were monitored for trichloroethylene and other volatile organic compound exposures extensively. And following this monitoring period, blood samples were collected at the end of their shift. We received the blood samples and performed our untargeted small molecule profiling to see what were the chemical signals and metabolite signals we could detect in these samples, and to see which ones were associated with the exposure, with trichloroethylene exposure. In addition to the measures that we were generating, there were other independent laboratories who were also measuring additional biomarkers in this population. These include immunological biomarkers, kidney injury biomarkers, as well as urinary exposure biomarkers, which were used to evaluate exposure classification. So once we performed our untargeted analysis of the samples, we performed a metabolome-wide association study, which is where we essentially tried to identify which metabolites we were detecting in these samples that showed a dose-associated change with trichloroethylene exposure. And when we completed our MWAS, we found that there were about 188 features that were associated with our exposure at our false discovery rate threshold. When we looked at what these compounds were, we found some very interesting findings. In addition to identifying biological or endogenous metabolites that were indicative of the potential effects of this exposure, we were able to find a large number of chemicals that we knew were chlorinated based upon certain features we could measure on those compounds, but we did not know what they were, and they did not match any of the known trichloroethylene metabolites other than a few of the common ones. So we decided to investigate these a little bit further. And to do this, we looked at how the different metabolites we detected that were associated with trichloroethylene exposure could also be linked to additional biomarkers potentially affected by trichloroethylene exposure. So when we performed this correlation analysis, we found that the known trichloroethylene metabolites that we detected in blood, these include trichloroacetic acid and trichloroethanol, were strongly and positively correlated with urinary TCE biomarkers that were measured in a separate laboratory. This was not surprising. This is what we expected to see. If these were not correlated, we would have been very concerned about the quality of our data. Where things became interesting is when we started looking at how these other chlorinated compounds, which did not match known trichloroethylene metabolites, were correlated with these other biomarkers. So we found that some subsets were negatively correlated with the immune biomarkers, which suggest immune suppression, which is consistent with what we know about the toxicology of trichloroethylene exposure, as well as a number that were positively associated with renal damage biomarkers, which again is consistent with what we know about toxic effects of exposure to trichloroethylene. So what this suggests to us is that these unknown chlorinated compounds, which were strongly correlated with TC exposure and also linked to the outcomes related to trichloroethylene exposure, are potentially novel or unknown TCE metabolites and provide one possible mechanism through which toxicity of these compounds could be exerting itself. And so as we are kind of following up on this study and continuing with this work, we're trying to determine what these metabolites are and running some model and in vitro studies to try and identify how these are linked to the outcomes that we know are associated with trichloroethylene exposure. So there's been a lot of evidence supporting these untargeted approaches and their advantages to understanding exposure and health outcomes. So our goal has really been to expand our ability to systematically apply these untargeted methods to human samples and human populations to better characterize the exposome for understanding environmental risk factors of disease. Our first goal, or one of our first goals, was to establish what we call an omic scale biomonitoring platform for better measuring exposure biomarkers. The results from this assay or the findings from this assay were recently published in Nature Communications. And what this allowed us to do is essentially create a relatively high throughput and comprehensive platform that has greatly expanded our ability to measure chemical exposures. And we're doing this by using a newer type of platform or mass spectrometer platform that has improved sensitivity towards these exposure biomarkers. At the same time, we're continuing to apply our more traditional metabolomic methods to exposome research projects. This is an example of more recent application to study benzene exposure, again, due to occupational or workplace exposures. And the advantage of applying these methods is that we have this ability to both look for metabolites of many of the exposures we're interested in, as well as characterize biological response or bioeffect profiles that we can now start examining for relationship to disease outcomes. So taken together, we have essentially a comprehensive platform and framework that allows us to provide sensitive and systematic measures necessary for exposome-wide association studies. We have the ability to characterize a very wide range of exposure biomarkers using untargeted methods. So we can use that to prioritize which exposures we should be studying, as well as the ability to understand the biological impact of those exposures and how those could be related to disease processes. So now what we've really tried to do is to implement this in a framework that allows essentially population scale or omic scale screening of the exposome with the end goal of establishing a cumulative resource for exposure disease relationships. So we know that we can use these untargeted platforms to both characterize exposure and disease effects. So our goal is to establish a reproducible, robust, and fair exposome analytical framework that allows us to do large-scale untargeted profiling and start assembling exposomes across different populations and across different diseases. And so to do this, we essentially have four goals kind of driving this, or four aims. So our first is to establish or to finalize our robust and large-scale profiling approach for the human exposome, where it's actually feasible to characterize thousands or tens of thousands of samples. While doing this, we want to do it in a way that improves the accessibility of our data for environmental health and biomedical researchers new to exposomic research. Oftentimes, there's a pretty steep learning curve to working with our data. We want to really try and make our data as open and as accessible as possible. My ultimate goal and focus in this is really this idea of a cumulative exposome cancer atlas. So how can we look across these different cancers that we know have an environmental component and start assembling similarities and differences in the exposure profiles linked to those diseases? And finally, how can we use the bioactivity relationships we identify to start prioritizing key exposures? And so for the next few slides, I'll really focus on more of the analytical aspects of what we're doing to make this concept of being able to analyze or perform large-scale exposome screening a reality. So one of the challenges we have faced is that our sample throughput, the time to do that essentially scales with the number of samples that you're trying to analyze per day. And so as you go from, say, 20 to 40 to 80 samples per day, if you're performing everything manually, it takes more time to complete those, and eventually you reach an upper limit where it's not possible to prepare enough samples to support your throughput on the instrumentation. So to overcome this limitation, one of the things that we've really focused on in my laboratory is automating all steps of our sample prep for untargeted exposome profiling. We've been able to successfully complete all steps of optimization, and we now have this workflow incorporated into our laboratory. And this was really thanks to some excellent work by Brooklyn McNeil, Prismar Pinto Pacheco, and Catherine Dunn, who are members of our research group. And the advantages of automating the sample prep is not only can we prepare a larger number of samples per day, but it also allows us to scale to meet demand for the number of samples that we have to prepare. So as we continue working on studies that are getting larger and larger, the methods that we've established on our robots can be easily implemented in additional ones that we purchase. We've also found that the use of robots really minimizes operator effects. And because the systems are so robust and consistent, we're seeing 15 to 20% improvements in our coefficient of variations compared to manual preparations. So this has been a huge amount of work that went into this optimization, but this has really allowed us to maximize our ability to run large studies and prepare as many samples as is feasible in a day. At the same time, we also want to have the ability to comprehensively detect as many different compounds as possible. So in addition to the automation that we've implemented in the laboratory, we've also gone through and systematically optimized the different analytical methods that we're using. In addition to expanding the analyses that we're performing, which allow us to detect more diverse metabolites, we're also including a framework for quantification of known compounds. So within our untargeted analyses, we're not only able to screen for unexpected or uncharacterized exposure, but we can actually quantify many known compounds within our data. And as part of this, we're now participating in proficiency testing. So we recently participated in the AMAP proficiency testing for PFAS biomonitoring, in which we passed all 3 levels in 9 out of the 10 compounds, showing that with these untargeted methods, we're not only able to screen for a very large number of compounds, but we can also accurately and precisely quantify these levels of known exposures. One of the challenges that we face in untargeted analysis is compound annotation. The mass spectrometers that we use don't tell us what we're measuring. Rather, they give us a mass and a charge of the compounds that we're detecting. And with that information, we have to try and extrapolate or predict what the actual compounds are that we're detecting. And so in order to facilitate this, we've really been trying to implement a standardized and routine annotation workflow that empowers our collaborators or assists our collaborators in taking advantage of much of the data as they're interested in working with, but also being able to just rely on or just resort to working with our very high confidence identified compounds. And so we now have a comprehensive computational workflow that allows us to generate the full unknown tables. We have systems in place for MSMS and annotation of our data, as well as annotating confirmed compounds, which is where we're comparing the data from our samples to standard databases and providing these high confidence annotations. And through much of our work, we're attempting to try and support large scale chemical identification. And we're doing this by creating standard libraries of compounds that we've run on our system. So we know that when we detect that compound in a sample, we're able to say, this is an amino acid or this is an exposure biomarker and provide an actual identity for that compound. So to date, we've run over 1,200 confirmed standards across our different assays. Many of these are endogenous metabolites, but we also have a large number of environmental chemicals, including over 200 pesticides, and a very unique library of 142 PFAS, which we were able to obtain from the EPA. These are PFAS that don't have authentic standards available for purchase, but are rather were collected from different companies and are PFAS used in industrial applications. We're continuing to grow our chemical databases. We have the entire ToxCast phase 3 library available in our laboratory. That will allow us to provide confirmation for almost 4,700 environmental chemicals and drugs. We're expanding our ability to confirm microbiome and phytochemicals, and we have additional endogenous metabolites that will be running on our platform. So in total, that will give us a database of over 7,300 unique chemicals representing many different exposures, drugs, commercial products, and endogenous metabolites on all of our assays. And so this will support rapid identification of thousands of chemicals in exposome studies. Again, one of the main challenges we face is identification and annotation of our data. And our hope is to create this as a research resource that can be used for really improving our ability to identify compounds in our data. And of course, we want to make this as operational and accessible to our collaborators as possible. So right now, we're uploading all of our confirmed chemical databases to GitHub. This is still private, but we're hoping to make it public very soon once we're done with data cleaning. Unfortunately, we're kind of exceeding what we can do at GitHub, and our plan eventually is to create relational databases where we have our confirmed compounds available to collaborators and other researchers. So it'll allow you to not only look at what's been confirmed and what you may or may not have detected, but also the standards we've run that are not detectable on our platform, so you can remove false positive annotations. And so that's really been kind of a quick overview of how we're improving the methodology for exposome profiling. And it's kind of important to think of where are we now and how have these actually helped us with exposome research. Our lab is relatively new. It was started in 2018. We had construction delays and then COVID delays. And so really we've been operating for about a year and a half or so using these standardized methods. And in that time alone, we've been able to analyze over 10,000 exposome disease studies, ranging everything from liver disease to nutritional intervention through World Trade Center first responders and occupational exposures. And our goal is to ultimately take this data and create these cumulative exposome resources where it will be possible to look across studies, across exposures, and start linking similarities and differences in exposome profiles across these different outcomes and populations. And so finally, I'll just give a very quick overview of how we're leveraging these analytical methods we're developing and the instrumentation we use for exposome research to better understand exposure to microplastics in human populations. So I'm sure I don't have to go into detail to really convince you of the challenge we face with global plastics production and pollution. But what's been really incredible to me is just the realization that there's been over 8 billion plastic products produced in the world since the 1950s. While some of these are recycled and reused, the majority of these plastics are essentially single-use plastic materials, which are discarded and placed in landfills. And so this creates a tremendous challenge from an environmental perspective, and also creates a scenario where there is a high probability of exposure, and plastics themselves are a very widespread pollutant in the environment. And the challenge we face in terms of exposure assessment and exposure monitoring when it comes to plastics is just the huge amount of compounds and classes of plastics that are used across many different product categories. This recent review by Helen Wessinger attempted to add up the different types of compounds that are used in the plastic production industry and found that there are over 10,000 plastic-related chemicals that are used throughout the plastic industry in the manufacture of plastic materials. So if we think of trying to assess exposure to plastics as a whole, it is almost impossible to think about measuring these exposures and understanding their pollutants in the environment. In addition to that, while you have these individual compounds, you also have the plastic material itself. which when it's introduced into the environment undergoes a number of different degradation processes where it's essentially converted or forms these very small plastic particles, which we refer to as micro and nanoplastics. And these are becoming widely recognized as a growing public health concern. These microplastics have been measured in pristine mountain catchments, suggesting that atmospheric transport and deposition is possible. They've been measured in both raw and treated drinking water. Microplastics have been detected in a variety of human samples, including feces and in lung tissue of individuals with lung cancer. It's been found in pet food and pet feces from across the United States, and most of even greater concern is many of these microplastics have been found in infant and adult feces. So we know that exposure to these microplastics is widespread and ongoing, but unfortunately it becomes very challenging to assess these exposures and try and understand how widespread they are in populations, because it's very difficult to measure these very heterogeneous plastic-related compounds and microplastics. And so we're focused on establishing what we refer to as an exposomic framework for understanding potential health effects of microplastic exposure. So to do this, we're using a multimodality strategy to measure different characteristics of microplastics in biological samples. These include mass-based measurements where we're trying to quantify levels of microplastics in biological samples. We're also attempting to characterize the morphology, including size and shape of microplastics in biological samples. The other thing we're interested in is compartmentalization. So for certain microplastic particles of different sizes, do these preferentially accumulate in different tissues or different regions of a cell? And also mobilization. These microplastics have very high surface areas, and they've been known to act as co-transporters of other environmental pollutants. So what are some of these compounds or co-exposures being transported by these microplastics? And our goal is to implement this characterization within a framework that also allows us to assess biological response to better understand potential health effects of exposure to these particles. And I know I'm running short on time a little bit, so I'm not going to go too into detail on the actual analysis that we're doing, but we have established multiplexed micro and nanoplastic measurement techniques that allow us to characterize different aspects of microplastics in biological samples, including the particles themselves. So what are the particle concentrations or the total mass of the particles, as well as free plastic additives and other compounds linked to the plastics. We're doing this through untargeted high-resolution mass spectrometry methods. So this gives us that ability to, again, characterize a very wide range of different plastic exposures. And our first projects are really focused on measuring the early life plastic exposome. So we're working with a number of different biological samples that are collected from the mother during pregnancy, as well as birth tissues, including placenta and amniotic fluid and cord blood, to better understand in utero exposure to microplastics. And we're doing this within a much larger study where the overall goal is to establish a toolbox for risk assessment of micro and nanoplastics in human population. Our ultimate goal is to identify what the key measurements and metrics are of microplastic exposures, so we can establish population level biomonitoring strategies that will allow us to better understand how widely these microplastics are distributed across the population. So in summary, there's really this need to establish improved measures of exposure and effect for determining the role of the environment, mainly the exposome, in disease. Combining untargeted high-resolution mass spectrometry strategies with automation and improvements in the methodology really allow us to now perform large-scale exposome epidemiology, where it will be possible to characterize exposures and biological response in thousands or tens of thousands of samples. And we're also implementing these exposome analytical frameworks to now start biomonitoring and study emerging exposures with our primary focus on microplastics, but we're also doing a lot of work with PFAS and other fluorinated compounds. So I'd just like to acknowledge my fantastic team. They've worked very hard on a lot of these results and generating these methods, specifically Brismar, Brooklyn, and Catherine for all their work on the automation and the sample prep, Xingping for his work on the LC methods, and Zoe and Anna for really all of the work that they've done establishing our microplastic methods. And of course, I'll have to acknowledge funding from NIEHS and the European Commission, who is supporting many of these projects. And thank you for your time. I'm happy to answer any questions.
Great. So this is an excellent presentation. And so microplastic is a very interesting topic that you recently did. Okay. So while we are waiting, the attendee enter their question to the chat box, chat or Q&A. So let me start one question. So, as an epidemiologist, so, our interest is, whether this, any like a target chemical associated with a specific disease. So that's why we are doing this conventional approaches. We based on the biology or any prior literature, and we measured specific chemical, and then in a population, and then link to the certain disease. But so it sounds like this exposome can measure many more chemical, and then doesn't need to have any a specific hypothesis. So, and then can allow, so that approach can allow doing a lot of these different types of environmental epi studies. So the question is, so then should we now move to this exposome, and then we don't need to conduct any of this conventional exposure assessment and then targeting chemical, but this exposome allow us to even not only identify this chemical, but also quantify the exposure level. So that's why this exposure can be a future direction of environmental epidemiology. So can you make some comments?
I see targeted and untargeted analysis as two very complementary approaches, and they're both important for understanding exposures and risk factors of those exposures. So I see the role for untargeted exposomics to really screen and identify what are the key exposures we should be studying to see how they're related to disease outcomes. So we know, about 80,000 chemicals are registered for use with the EPA. Those can form any number of different breakdown products or metabolic products. And so having the ability to screen for a very wide range of exposures is important to kind of prioritize what are the key exposures we should be studying. That being said, once we identify those relationships or have candidates in mind, the targeted biomonitoring is very important because it establishes quantifiable measures that have a much higher degree of confidence in terms of the accuracy and precision, and they're much more appropriate for risk assessment, especially if you're going to be using any of that data for regulatory purposes. So I see the exposome as a means of prioritization, and then the targeted methodologies is really what gives us the ability to test our hypotheses on how those exposures could be linked to that disease.
So then, does that mean that this exposome does not necessarily provide information about actual quantity, so the exposure concentration for specific chemical, but this is more like a screening this many different chemicals and then prioritize which chemical we need to focus. So once we conduct this exposome study and then identify those chemicals, then so next step will be more targeted approach, and then using the conventional approach, and then measure the concentration.
Yeah, so in order for us to quantify an analyte that we're detecting, we need to have a standard for that compound. And oftentimes that standard is not available when we're performing our screening. So we add PFAS and a number of other exposure biomarker internal standards to our samples when we're preparing them. And then we also run calibration curves, which allows us to quantify those known exposures. But if we don't have those standards, we can't generate actual concentrations. That being said, our data is quantitative in the sense that you are measuring a intensity that's proportional to concentration. So I know in most cases that if I'm looking at two samples and the intensity I'm detecting in sample A is lower than the intensity in sample B, I can say that sample B probably has a higher concentration, but I can't tell you what that actual concentration is in terms of, say, nanograms per mil.
Okay, so we have a question from the audience. So what are your thoughts on developing preclinical models for exposure-linked research with relevance for humans?
Okay, so I'm going to take a stab at that, but please correct me if I'm not understanding your question correctly. So when you say preclinical models, are you referring to establishing like a in vitro or animal model where we are studying exposure outcome relationships we see in humans? Is that correct?
I think so.
Yeah. I don't know if they can unmute themselves. Maybe they can't. So
yeah.
Yes. Yeah. Okay.
Human, like, yeah.
Perfect. Yep. Yeah. Absolutely. So that I see is one of the advantages of using these approaches, because it allows us to essentially start developing systems or informed hypotheses about exposure disease relationships in humans. So what we're measuring when we're working with the population study is a correlation. We can't say anything about causality from our data. We're measuring an exposure. We're identifying metabolic relationships to that exposure and how it's linked to an outcome. So all we can do is really use that data to start forming our hypotheses about potential mechanisms. But with those hypotheses, and once we start forming them, we can start designing the in vitro or the mechanistic studies that will allow us to start understanding what the potential relationship is between that exposure and the outcome. That's very much what we're doing in our TCE study, where we found these novel TCE metabolites, and we found these associations with immunological and renal damage biomarkers. So if we start, incubating our exposure with, S9 fractions or different cells, can we generate these? Do we see any effects of these exposures? So yeah, it's a very powerful tool. And the advantage of doing the preclinical models is that, you can identify a relationship in a population. If you can then explain that relationship within an in vitro or an animal model, it's very, very powerful. And another interesting thing that we've noticed is that when we apply our platform to primary human cells, one of the things that we've been really fascinated by is we can use our metabolomic profiles to actually distinguish differences in response between who that cell donor was. So, for example, when we have primary cell lines from male and female donors, we actually see very large differences between how they respond to different exposures or different culture conditions, which allows us to essentially phenotype our system even further.
Okay, so one last question. So when you measure plasma POPs, do you simultaneously measure lipid level for POPs adjustment, or do you just present POPs data unadjusted for lipids?
So the challenge is that the traditionally reported lipids for POPs data is not something that we would be able to measure on a mass spectrometer. When they do the POPs measures, typically they're measuring total cholesterol and total triglycerides. That's done using different assays. We can't really sum up and generate those measures from the mass spec. So it's not something we measure in our data. Depending on the study, we can measure them. Oftentimes we find that lipid adjusted and unadjusted data is so highly correlated that it's not really necessary to generate the lipid levels. Usually the correlation's about 0.9 to 0.95. But if there's a need for it, or if we need to have big differences in BMI, then we will measure it. But typically we don't unless it's requested.
Okay, so this is 12:52, and then so even actually I have a few questions, but we have to end this session. So thank you everyone for joining this seminar, and then thank you, Doug, for your this excellent presentation. So we will have a next the research and resident webinar next week, and then so we will, so the Merides will announce that webinar by email. So thank you for joining this webinar, and have a wonderful day. Bye-bye.